Industry skills, practical knowledge, real-world impact. This is Simple Gyan.
What if I told you that a sticky black waste from a sugar factory can become acetic acid, the same chemical family used in vinegar, food preservation, textiles, adhesives, paints, and chemical manufacturing? Here is the surprising answer. 1 ton of sugar-rich molasses can theoretically hide hundreds of kilograms of acetic acid potential, depending on sugar content and process efficiency. Molasses is not waste. It is a concentrated carbon source. Molasses is the thick syrup left after sugar crystals are removed from sugarcane juice. It contains water, sucrose, glucose, fructose, minerals, ash, nitrogen compounds, and organic acids. In one reported sugarcane molasses composition, sucrose was about 36 g/100 ml, glucose about 9 g/100 ml, fructose about 11 g/100 ml, water around 20 g/100 ml, and ash around 7 g/100 milliliter. This sugar is the fuel for microorganisms. The process happens in 2 biological stages. First, yeast converts sugar into ethanol. Second, acetic acid bacteria oxidize ethanol into acetic acid in the presence of oxygen. This is the same natural principle by which alcoholic liquids turn sour when exposed to air. Acetic acid bacteria oxidize ethanol to acetic acid under aerobic conditions. Raw molasses is too thick and too concentrated for direct fermentation, so it is diluted with water to bring sugar concentration into a suitable range. The plant removes suspended impurities by screening or filtration. Then pH is adjusted, nutrients may be added, and the feed is sterilized or pasteurized to reduce unwanted microbes. Now yeast, usually Saccharomyces cerevisiae, converts sugar into ethanol and carbon dioxide. Reaction: C6H2O6 2C2H5OH plus 2CO2. The theoretical ethanol yield from glucose is about 0.51 kilograms ethanol per kg sugar, but real plants get lower yields because some sugar goes into yeast growth, byproducts, and losses. Engineering control: temperature, around 30 to 35 degrees Celsius, pH, around 4.5 to 5.0 time, typically 24 to 48 hours. Main product: fermented wash containing ethanol. The ethanol-rich fermented wash can go in 2 directions. In one route, ethanol is distilled and concentrated before acetic fermentation. In another route, the clarified alcoholic wash is directly sent for acetic acid fermentation. Distillation gives better control, but it consumes more steam. Direct fermentation saves energy but requires better microbial control. This is the heart of the process. Acetic acid bacteria such as Acetobacter oxidize ethanol into acetic acid. Reaction: C2H5OH plus O2CH3COH plus H2O. This reaction needs oxygen and produces heat. That is why aeration, agitation, and cooling are critical. The theoretical yield is about 1.30 kilograms acetic acid from 1 kilogram ethanol, based on molecular weights. The process is aerobic and exothermic, so oxygen transfer becomes one of the most important design factors. Here is the engineering challenge. Bacteria need oxygen, but too much aeration can strip volatile ethanol and acetic acid from the liquid. Too little oxygen slows the reaction. Too much air increases evaporation losses and foam. So the plant must balance airflow, agitation, temperature, and antifoam dosing. Main controls: dissolved oxygen, temperature, acidity, ethanol concentration, foam level, airflow rate. Let us take a simple example. If 1 ton of cane molasses contains roughly 50% fermentable sugars, that means about 500 kilograms sugar. Theoretical conversion: 500 kilograms sugar times 0.51 is equal to 255 kg ethanol. 255 kilograms ethanol times 1.30 equals about 332 kilograms acetic acid. But industrial biological processes are not 100% efficient. After fermentation losses, side reactions, evaporation, cell growth, and purification losses, practical output may be much lower. This is why plant design and process control decide profitability. After acetic fermentation, the broth contains acetic acid, water, biomass, minerals, color bodies, and unreacted compounds. For vinegar-grade product, the liquid is clarified, filtered, standardized, and pasteurized. For higher-strength acetic acid, water removal becomes difficult because acetic acid and water are strongly associated. The plant may use distillation, extraction, dehydration, or membrane-based concentration depending on purity target and economics. Key point: making dilute acetic acid is easy. Making high-purity glacial acetic acid is the real engineering challenge. Acetic acid is used in food-grade vinegar, acetate salts, textile processing, cleaning products, chemical synthesis, and as a raw material for acetate esters and vinyl acetate-based products. Fermentation-based acetic acid is attractive because it uses renewable biomass. But for bulk chemical markets, cost, separation energy, purity, and productivity decide whether it can compete with petrochemical routes. So the journey is simple in concept but complex in engineering. Molasses gives sugar. Yeast makes ethanol. Acetic acid bacteria oxidize ethanol. Separation technology purifies the final product. And that is how a dark, sticky sugar industry byproduct can become one of the world's most useful organic acids. The surprising truth is this: the real factory is not just steel tanks and pipes, it is a controlled ecosystem of microorganisms, oxygen, heat, and chemistry working together. Endline: from sugar waste to industrial acid, this is biotechnology inside chemical engineering.